Good afternoon, everyone.
Good afternoon.
Welcome to Data Blitz. I hope you continue to be safe and well. Now that the spring semester is over, you're all in a relaxed mood, enjoying coffee or whatever your favorite drink is. We are in for 6 presentations. And if you are new, let me briefly outline the guidelines for this Data Blitz. The event is aimed at generating collaborative interests and also make us appreciate what a wealth of knowledge and expertise we have right on our campus. So it gives us all an opportunity to get to know various research projects that are in progress. And also it gives us an opportunity to find some mutually beneficial collaborative grounds, because these days complex problems face us, and complex problems require complex solutions and hence collaborative efforts. So before going further, let me review the guidelines. Each presenter is allowed 8 minutes, and I will let them know when 1 minute is left. And attendees are encouraged to ask questions through the chat box, make suggestions, or even approach them for exploring collaborative interests all through the chat box, and the presenters will get back to you by email. So, by the way, this event is being recorded. Later on, all these recordings will be posted and made available on our website so that you can go back and look at any of the events that you have missed, or events that you have attended but still want to review. Now, without further ado, let us get started with our first presenter, Dr. Sameh Abdelwahed. Dr. Abdelwahed,
Yes.
are you there?
Yeah.
Please go ahead.
Okay. So good afternoon, everybody. Thank you, Dr. Chris, for giving me the opportunity to talk about my research. So my name is Sameh, and I'm assistant professor in chemistry department since September 2018. As you can see here, I have a good experience in many chemistry fields like organic chemistry, bio chemistry, medicinal chemistry, enzymology, supramolecular chemistry, and nanotechnology. So basically today, I'm going to talk about 3 projects, as you can see here. The first 2 projects are very related to each other, and I'm going to just try to make my language is understood for everybody. So I'll try to avoid the scientific language to make it easy. But please, if you have any question or anything, you can write it in the chat, or you can email me. And I will be really happy for any collaboration with any of the medicinal chemistry projects which I'm going to talk today. So the first 2 projects actually is very related to each other. So they are talking about drug delivery. Okay, so the first 2 projects is drug delivery. The first project is using vitamins as a vehicle to deliver the anti-cancer drug to the tumor cell. So the first project is using vitamin as a vehicle to deliver the anti-cancer drug to the tumor cell. So basically, as we know, the main difference between the tumor cells and normal cells is that tumor cell is keep growing without limits, while the normal cells has something called apoptosis. Apoptosis is programmed cell death. which is really simple to just say, like, while I'm talking to you, there are many hundreds of cells is dying in your body, and many of them is growing, but the cancer cell is just keep growing. Okay, so because it's keep growing, so it need more nutrition, it need more food, it need more vitamins. Okay, so the vitamins which I'm going to talk today is vitamin, which is vitamin B1, which is thiamine. So thiamine is a vitamin which is very necessary for the cancer cell. So basically, the idea is to link this. If you can see the figure number 1 here, here's the thiamine, and you just link the thiamine to the drug to make a link between the thiamine and drug. And by this way, you are going to make the cancer cell uptake more of the anti-cancer drugs. So thiamine here is going to work as a vehicle which is going to deliver this drug, which is, for example, in this figure here and the blue ball here, to deliver this drug into the cell. Okay, the next project, which is very close to this one, and I forgot to say the collaboration with Dr. Scanty. He opened his lab for us to do the biology part. So basically, we finished like 50% of this compounds, and we are doing the other 50, and we are going to check the biological effect of this compound to the cancer cell in Dr. Scanty's lab. The other project, which is also related to the drug delivery, is now we are using thiamine as drug delivery to link the thiamine to the antibiotics or the bioactive molecules. So it is the same idea. You are going to link the antibiotic to the thiamine. So the bacteria or the pathogenist is going to take the thiamine, thinking it is just thiamine, but it is thiamine with inside it is the drug or the antibody there. So the bacteria will take it, it will get released into the cell, which the bacteria cell, and it will eventually kill the cell. So in this project, we have a collaboration with Dr. Moore. Also, we are using his lab to do our biology part. The last project, which is basically it is talking about traditional medicines as treatment of the COVID-19. So basically, the advantage of the herbal drug or the traditional medicine is it is safe, and you don't need to take any FDA approval. So upon discussion with Dr. Grass, we select 3 plants, which is really growing good in Texas. And the 3 plants which we choose is Artemisia and Rosales, or the Hibiscus, and the ginger. So in these 3 plants, just to make sure it is COVID-19 active. So basically, because you can't do biological work with COVID-19 in our university at least, so the basic idea is to download the X-ray structure of the COVID-19. It is available online, and you can download it. And so we have these 3 plants. So I'm just going to talk about ginger as an example, which I'm showing here. So in ginger, there are many compounds. There are like tens of compounds in ginger. And to know if it is going to be active for COVID-19 or not, so we have all these compounds, and we do something called docking. So basically, you download the COVID-19 and structure, and you have the structure of your compounds in ginger, for example, and you are going to make scanning to see which compound is going to be active. So to make the story simple, is just imagine yourself as COVID-19, and you want to enter your room, the bedroom, which is the cell. So you are going to enter with your hand, like you are going to open the door with your hand. COVID-19 also has what we call active sites. This active site is going to open the door. So basically, we scan all these compounds and we make sure which compound is going to inhibit this virus. So if you can see in the figure number 3, you can find the compound which we choose from the ginger here, and the other structure is COVID-19. So basically, you are inhibit this virus here. Also, we have another. So you basically are inhibit the hand of the virus. Also, you can inhibit the virus to enter the door by locking the door. So we have another compound which is going to lock the door and make the COVID-19 hard to enter the cell. Okay, thank you very much. This is just a brief.
Thank you, Dr. Abdelwahed. Here is an excellent example of a synthetic organic chemist using his expertise to attack cells, cancerous cells, virus, and bacteria. And obviously, he has collaborated, or he has been collaborating with the experts in the right areas. And sometimes you can even look for a computational chemist. In docking experiments, usually you have a protein and a small molecule, and geometry is needed.
That's true, yeah. Actually, I have a very good postdoctor who is doing this computational chemistry.
Again, it opens up a door for others who may be dealing with other kinds of diseases, or even plant diseases, plant viruses, where it may be useful to look for friendly organic molecules. So this is something one may want to think of, agriculturalists or even people with the interest in animal systems, because the approach offers a lot of scope for exploring other areas. So I hope if you have any questions or any comments or any offer to collaborate with the Dr. Abdelwahed, please don't hesitate to let him know through the chat box. Again, thank you, Dr. Abdelwahed.
Thank you.
Our next speaker is Dr. Shuza Binzaid. Dr. Binzaid? Hello? Dr. Binzaid? Okay, we will go to the next one. Dr. Dong? Dr.
Yes.
Xishuang Dong?
Yes, I'm here.
Okay, please proceed.
Okay. Good afternoon, everyone. I'm Xishuang Dong, assistant professor from the Department of Electrical and Computer Engineering, and I'm very happy here to share my research interest and some areas, and hopefully, I mean, we can build some collaborations on research based on our research interests. Actually, my research focused on developing AI technicals to do data analysis so as to provide some critical information to make some decisions. Specifically, I'm working on building machine learning models and deep learning models to solve various problems. And later on, we will build different applications based on this research. Actually, there are 6 active research areas I'm involved in. The first one is I'm doing some research on COVID-19 data analysis. Actually, this research will cover different kind of data like the images and tests. And for example, actually, we try to recognize the COVID-19 disease from medical image like the X-ray and the CTs. Actually, you can see the figure 1. Actually, the just examples. We can recognize the COVID-19 with X-ray data. Actually, this research involves more than 10,000 medical images. It's a public data site. And in the future, actually, our goal is trying to build some like the multimodal COVID-19 data processing pipeline. Actually, this research will try to mimic the diagnosis procedure like doctor doing in the for the patient. Actually, when the doctor do the diagnosis for COVID-19, actually, they will try to make the diagnosis with different sorts of information like the image and the clinical trials. So finally, we will build a model to mimic this procedure. And in addition, I mean, for the biomedical domain, we also do some research on biomedical data, big data analysis. For example, actually, we try to do some research on single-cell sequencing data analysis. Actually, this research will involve many challenges like the limited ground truth and high dimensionalities of data and data sparsity. So actually, we propose different kinds of deep learning model, like the Sameh super learning model. And actually, you can see the figure 2. That's the framework of the Sameh super learning model. Actually, we have verified this framework on different problems, like the cell type identification. And moreover, actually, we built some model to do feature selection and dimension reduction to overcome the ACU, the challenges like the high dimensionality and data sparsity. For the research on deep learning theory and application, actually, I have mentioned, I mean, all my research will be based on, I mean, machine learning model and the deep learning model we developed. So actually, we make more efforts on to propose some novel machine learning model like the Sameh super learning model, transfer learning model, and ensemble deep learning model. And with this kind of methods, actually, we can build application like the fall detection on seismic data and load forecasting on, I mean, smart grid. And another interesting research area is about fake news detection on social media. Actually, today, actually, the COVID-19 pandemic is a global issue, and on the Twitter, actually, there is some misinformation spreading on the Twitter. And recently, the Twitter collaborates with a AI group, a research group from Stanford University to aid warnings to tweets with, I mean, misleading COVID-19. So actually, I think we can build some contribution to this area. Actually, we have some method to do pre-processing on the social media data. And we also can do the prediction of the fake news on the social media data with our own, I mean, deep learning model, for example, like the Sameh super deep learning model. And okay, finally, we also have two research areas. It's about the natural language processing and image processing. Actually, we do semantic analysis on tests and also test classification. And for the image processing, actually, we do image classification. Specifically, actually, we have proposed a distributed structure for image classification on IoT structure. And so in our words, I mean, actually, I'm focusing on the deep learning and machine learning model to process different kinds of data, and hopefully I can help some like the research group or faculties to process your data and with automatic methods. Okay, that's all my research from my side. Thank you.
Thank you, Dr. Dong. Are you responding to any COVID-19 proposal solicitation currently?
Yeah, actually, I'm planning to submit a NIH program. It's about developing some like the biomedical tools to enhance the detection of COVID-19. Actually, the due is June. Actually, it's NIH R21. Actually, the due is June 16. I'm working on this proposal now.
Are any collaborators joining you on this proposal?
So far, actually, I'm working with Dr. Kim. I mean, also, I have somebody have expertise on like the X-ray data or CT data. That will be very helpful for this proposal. You know, for this proposal, we have to understand this kind of data like the X-ray and CT. For example, for the figure 1, actually, from my opinion, actually, it show they are very similar, actually, frankly. So, but the expert from the, medical domain, maybe some doctor will tell me, okay, what's the difference between these two figures and how they get this disease, like how they get the image like this with COVID-19 disease.
Dr. Dong, during the first Data Blitz event, there was a mathematician who presented research interest in image analysis. Are you aware of that expertise on our campus? Dr. Agigi can respond.
Okay. I will try to connect to him, and hopefully, maybe we can have some collaboration on this point.
Are you looking for any collaborators in other areas? For example, could you collaborate with Dr. Abdelwahed, because he is doing some docking experiment? Would your expertise be of help to him?
Also, okay.
He's actually using an X-ray crystal structure of the virus.
Okay. Let me check the data first. You know, I think I can provide some suggestion on the methods, but before that, I have to understand the data and task first.
Just my curiosity.
Thank you.
Are you looking for any collaborators in other areas where your expertise can be of help?
Actually, I'm open to any collaborations, and actually, frankly, so far I'm just focusing on this proposal. I mean, the COVID-19 proposal. Okay. So I'm very happy to have collaborators with any infected. Yeah.
Okay. I just see in the chat box, Dr. Dada is interested in collaborating with you, so you may want to contact.
Sure. Me. Yeah. I saw that. Yeah. I will email to all. These interesting points. Yeah.
Thank you, Dr. Dong, for an interesting presentation.
Thank you. Yeah, it's my pleasure.
Again, attendees, if you have any problem that requires analysis of a huge amount of data from multiple sources, you know who to contact. You just heard that person. Okay, let us move on to the next speaker, Dr. Gururaj Neelgund. Dr. Neelgund?
Yeah, I'm here.
Please proceed.
Yeah, thank you, Dr. Place. Hello, all. Good afternoon. Okay, I would like to thank for Office of Research for providing me this opportunity to present my research here. Okay, I'm Gururaj Neelgund, Assistant Professor in the Department of Chemistry. Okay, I received my PhD in chemistry and got the postdoctoral experience in polymers and nanotechnology. And presently I have 50 journal publications, and I'm the associate editor of American Journal of Engineering and Technology and the Technology Research, and editorial board member of the current research in materials chemistry. My current research area is emphasized over the designing of nanomaterials for photothermal therapy of cancer and purification of water by photocatalysis. Okay, our key plan is to synthesize the photoactive nanomaterials or light sensitive nanomaterials that consist of carbon nanomaterials like graphene nanosheets, carbon nanodiamonds, and carbon nanotubes, with which other nanostructure materials like polymers, metal nanoparticles, metal oxide nanoparticles, and other metal chalcogenides like the sulfides or selenides. Okay. So for photothermal therapy of cancer, we prepare the near infrared active, that's called NIR active nanomaterials. The reason for choosing or preparing the NIR active nanomaterials for photothermal therapy of cancer is that if you use the NIR added therapy, this near infrared radiations are minimally absorbed by the body tissues. That is the reason we choose the NIR active nanomaterials for cancer therapy. So whatever the nanomaterials we design, those nanomaterials are able to generate the enormous amount of heat under exposure to near infrared radiations. That's called photothermal effect. By the process of this photothermal effect, it is possible to destroy the or kill the cancer cells in a selective manner without harming to the normal or healthy cells. Okay, so that is the one area we are working right now on that. And other area of research is the nanomaterials for photocatalysis for the purification of water. Okay, so if the waters are contaminated with carcinogenic colors, whatever it is used in industries called dyes, okay, those dyes can be degraded to the environmentally friendly materials like carbon dioxide and water using the nanomaterials as photocatalyst what we prepared under the illumination or irradiation to UV visible or sunlight. Okay, so whatever the nanomaterials we prepare, that is used for both the cancer therapy and the photocatalysis. Okay, that is the one. So some of the nanomaterials I would like to show here. Okay, you can see in the slides, these are the scanning electron microscope of some of the nanomaterials we prepared. Okay, the first image is the graphene nanoseeds. Okay, this shows the microstructure, the morphology of the graphene nanotubes what we prepared in our lab. And the second one is the image of the graphene nanoseeds and zinc oxide nanoparticles. Okay, you can see the both the graphene nanoseeds and rice grains shaped zinc oxide nanoparticles in the second image, whereas the third image shows the nanocomposites prepared by graphene nanoseeds and polyaniline nanotubes. So whatever the image you see in third image, that shows the whatever the tubes you can see there, those are polyaniline nanotubes. Those are deposited over the graphene nanoseeds. And the fourth one is the graphene nanoseeds and zinc oxide nanotrapezoids. Okay, those are the composites of those. Okay, you can see these are the two areas currently we are working in our lab. Okay, that is the one. So if anybody interested with collaborate for us or with us, you are most welcome to approach, and we are open to collaboration. Okay, and I would like to thank our department head, Dr. Oki, who has supported me to do this and helped me to do this research. Okay, this is all about my research. If anybody wanted to collaborate both on research and also the grant proposals, we are glad to do that one. Okay, that is the one. This is all for our research. Thank you all.
Thank you, Dr. Neelgund. Are you collaborating with Dr. Komalapati?
No, actually, but yeah, I know Dr. Komalapati, but we'll plan to do that one. Okay.
Are you looking for collaboration in other areas besides cancer therapy?
Yeah, right now we know the other biological activities like antimicrobial or antibacterial. Okay, these actually what the nanoparticles or whatever the nanocomposites we prepared, those have that activity also. Other biological activity like antimicrobial or antibacterial. Okay, so if anybody wanted to check with that, yeah, I'm glad to do that also. Okay.
Any interest in pursuing any COVID-19 opportunities using nanoparticles?
Okay, right now I don't know. I don't have any experience with that one because basically I'm a chemist.
Right.
But I don't have any biological experience with that. Right now also in cancer, right now whatever we do, okay, we are looking for any cancer, okay, expertise. I mean, who works on cancer cells. Right now, I don't know, I don't have any experience with working cancer cells, okay. If somebody want to check with these, whatever the nanoparticles or nanomaterials we prepared to further proceed, because we are publishing until whatever we measure the photothermal effect, okay. That is the one we are going to do now, okay, what I did so far, okay. If somebody want to further extend that one, we are glad to do that one.
Thank you. Again, attendees, please note Dr. Neelgund's expertise in nanomaterials, also photothermal effect, using that effect as a therapy. So anything that needs to be destroyed, perhaps you can think of it. Thank you again.
Thank you, Dr. Krish. Okay, thank you. Thank you all.
All right. Let us move on to our next presentation. I hear Dr. Binzaid is here, so I will ask him after Dr. Kim. Oh, sorry.
Okay.
Yeah, just
Thank you. I'll be.
Oh, you are there. Okay.
Yeah, I'm here.
Second,
yes. That's okay. I can go later on. I'm done most of my work today.
That's fine. Are you ready now?
Yes,
okay,
I'm
our
ready.
speaker now is Dr. Shuza Binzaid. Please.
Yes. Thank you. First, I'm sorry. Actually, I wanted to cancel. I had few things going on this morning. I did not know that I should be able to finish it, but I'm glad I finished it. In the meantime, Dr. Atia gave me a call. Where are you at? You are supposed to be presenting. I said, I canceled that program already. So anyway, I'm ready now.
Hear you.
Thank you. Thank you. Mostly here, I'll be talking about myself because I guess I owe everyone introducing myself. I'm fairly a new faculty here, a research associate professor. I came from UTSA. And I'm here at Prairie View. I'm glad I'm here at Prairie View for last year and a half, and I'm trying to push quite a number of projects. And my interests are in renewable energy. I'm currently appointed with Smart Center. So it's a CRI center, as you know. My interest also in my power electronics and energy conversion, and semiconductor microelectronic design, fabrication and testing, microcontroller systems, and multifunctional sensor devices. So, and also various renewable energy sources. So I have about 20 years of experience in various projects leading and innovative teams. I worked for almost 10 years in the industry, and also experience in various areas, research about 14 years altogether, and teaching experience also about 8 years. So I have innovated a number of technologies throughout these years, about 12 altogether, and almost 8 of them in a pipeline with US patents. So here at Prairie View, I have two good news here, talking about two new innovations already filed for patents, and I have to push myself to for the utility petal conversion coming end of this year. And other new areas that I'm looking at is thermal energy conversion to electrical energy. And I have a master's student working in this, which is figure 1 showing. In this process, we'll be using in a closed loop system, the thermal energy from sun will be converting a vaporized liquid that will have high enough energy to rotate a turbine. So then the turbine will be connected to the electric power generator. This is how we are planning for. At this time, we are making a small size of the entire system, which already has a couple of innovation in it. One of them is a self powered pump that is required to overcome its own pressure. So this pump is actually a set of valves that work against each other to pump the condensed liquid into the thermal chamber. The thermal chamber is the high energy vapor is produced in that. So that is probably we are looking at 2 innovation that will be trying to secure IP at Prairie View. The second one is almost ready for filing patent. In fact, one of the patent we can file, and we can file later in this year, and the second one. This is a multifunctional sensor. It's a radiation sensor at this time, but it can detect proton radiation, other includes like X-ray. We still need to do the neutron radiation probably sometimes next year. But because the electron and the proton test have been completed, so we can tell that neutron definitely will detect on it. So it has a characteristics of detecting radiation. It has been proven. Later on, we have proved that this sensor can detect air ionization. So that includes like a thunder strike. So pre-thunder strike, we can detect with it. So that's another built into this sensor. And the third one, we are trying to do the electromagnetic wave, especially the cellular tower energy emission. And we already have tested with our mobile phones at a very close proximity. And our preliminary test, we still have to do more. Preliminary test shows that we can detect the ionization in the air due to the energy transmission. So those are the things coming out of a single. We have redesigned our sensor. So the figure 2 is showing the sensor, and the side is showing 2 Arduino microcontrollers actually acting as a parallel computing. We developed the program for it so it can detect. So in future, we'll be seeing a lot of modular systems coming out with a small sensor with just a Arduino microcontroller on it, which can people can use as a badge, and it can detect any radiation or the thunder strikes. It can detect ahead of time for the thunder strikes. So hopefully we'll see few of these devices in the market soon after we secure our IP. That's all I have.
Thank you, Dr. Binzaid. Do you have any collaborative interest with Dr. Wilkins?
Yes, I'm actually, Dr. Wilkins is working on carbon nanotube. Dr. Marlin is laughing because she's working with Dr. Wilkins also. Okay, so yes, I'm actually guiding one of his students also, getting some data collection, certain ways of doing it. I have programmed microcontrollers for Dr. Wilkins also. And last time we went to Los Alamos lab, we did not take our big 50-pound weight instrument because I had this microcontroller with me, and I made the microcontroller for him also for his test. So he just went, it's a pocket portable thing, and we just went over there and did our test successfully. Yes, I'm collaborating, helping him the test procedure with his carbon nanotube, and helping the student on the side for this his project. And I have a plan now. Once we get back to campus, I have a plan that I will actually sandwich my sensor with his carbon nanotube. So it can be a hybrid, multifunctional sensor for the future. We can do total ionizing radiation dose, plus I have the single event detection. So that can be another generation of sensors in future that I'm looking at.
If you are asked to expand your collaboration, what areas will you be looking for?
Oh boy, I'm looking at many different areas now. I need to make
any biological studies would be benefited by your.
Well, biological studies would be the radiation detection for human safety. Obviously, it is one of the criteria I'm looking at. So, do you have anything? Anybody with a lot of money, sitting pile of money? I can use some.
I can direct you to Dr. Sargenti because I don't know about money, but I know about his research interests. So,
Dr. Sargenti has actually we met once, and because one of his student, Mr. Raman, was there with us for photo detective under radiation.
right.
So, so he was there, and Dr. Sargenti came to find out also from the student that I have a device now, testing. So he showed his interest also.
Okay. Participants, please direct any comments or questions to Dr. Binzaid. He is extremely enthusiastic with his inventive efforts. Please contact him through the chat box. Again,
sure.
Thank you, Dr. Binzaid.
Thank you so much.
Our next presenter is Dr. Seungchan Kim. Dr. Kim.
Hi, can everyone hear me? Okay.
I can hear you very well.
Okay, great. Okay, so thanks, Chris, for inviting me to be able to talk about what we do in our center. So I'm going to mainly talk about what we do in our center, not just focusing on what I do in terms of research. So I am leading our Center for Computational Systems Biology at Prairie View. We started the center about 3 years ago, and you can find more up-to-date information on the website address that I put up on top of my slide. So you can always come there and then take a look what we are doing in terms of research and papers and others. So our research is centered around systems biology with the computational tool, heavy focus on development of computational tools. We do a lot of mathematical modeling, and we also do a lot of computational analysis. And I think Xishuang just talked about what he does in our center in terms of big data analysis, and then recently on the COVID-19 related research, and then single cell RNA sequencing data analysis, and so on. And our main focus at this point is study of cancers and neurological disorders like a Parkinson's disease. But we also want to expand our research area into like agricultural research and then animal science and so on. So I'm going to talk briefly about what we do in that area. So today I'm going to focus on a specific research project that we are currently have ongoing. So the first, I'm going to talk about extramural research project, meaning all of this research in collaboration with our extramural collaborators. And all of this, most of those research are currently sponsored by either federal government or others. So the first project that we have is a stand-up pancreatic cancer research stream team. So basically what it is we are part of a big international consortium where we are studying new novel therapy for pancreatic cancer patients, mostly on the clinical trials. So the goal of this project is to launch and complete and there's 3 different clinical trials based on the chemotherapy or other treatment options for pancreatic cancer with patients. So it's a pretty active and large consortium. We play a very small role in the research, mainly providing bioinformatics and computational support for whatever the data they generate from the research. So that's one of the project. Second project is about biomarker discovery, what we call exosome biomarker discovery for pancreatic cancer. This is sponsored by NCI, Early Detection Research Consortium, collaboration with City of Hope. It's a big National Cancer Center in California, near LA, and the Translational Genomics Research Institute and others. So, and then we have a lot of, and there's quite a few hospitals and clinical centers participating in this to provide patient samples so that we can do genome RNA sequencing of all of those cancer patient samples and then do the biomarker discovery. So our role in this project is to do computational data analysis to discover potential markers for early detection of pancreatic cancer. In pancreatic cancer, early detection is very critical because most of the pancreatic cancer are detected or diagnosed at the very late stage where the treatment option is very limited, and then the survival option, the rate is really, really low. So in this project, we are trying to use less invasive option like a blood, and then do the sequencing, and then try to identify the markers for the pancreatic cancer detection. So in other words, if we are successful, we should be able to develop a biopanel that the clinician can use and then tell you, hey, you have a pancreatic cancer or early indication of pancreatic cancer. So far, this is the second year of the project. We just got the sequencing data from City of Hope. So we actually get the data, and then we actually do the processing here. So I just downloaded, we just finished the processing of about 1,500 cancer patient sample. It's all on our workstation, and then we process them, and then we're just about to back up those data outcome to the City of Hope, and then we will do the further analysis to do the biomarker discovery. So that's the second project. Third project is sponsored by DOD. It's a collaboration with the Johns Hopkins Medical School. It's a multiple PI project, meaning Johns Hopkins and the Prairie View play an equal role in this project. So I am one of the PI, and then there's another PI at Johns Hopkins. So Johns Hopkins will be responsible for collection of patient sample, and we are responsible for sequencing of those patient sample and data process, data analysis. So in our center, we actually have a high-throughput next-gen sequencing machine available. And in this project, we're actually getting the samples, and then we will actually do the sequencing and then data analysis. So that's a third project. The fourth one is biomarker discovery. As you can see, we do a lot of biomarker discovery for the Parkinson's disease, sponsored by Michael J. Fox Foundation, and then you know the reason. And then this is a 2-year project, and then we are we just finished the first year, and we have about, again, our role is mainly computational, and the data are being generated by my collaborator at TGEM, at Translation Genomics Research Institute. We just got the data, all of actually close to 5,000 patient samples sequenced at TGEM, and all those data are transferred to us, and then we are doing the analysis. Actually, Xishuang Dong, who just presented, he's actually doing the data analysis for that project as well. Third one is chemotherapies for cancer to pulmonary hypertension. In other words, there are lots of chemotherapy options available for the cancer patient, but not much on the pulmonary hypertension disease. So we are collaborating with the University of Pittsburgh Medical Center, and then trying to see whether computationally, whether there is a way to repurpose chemotherapy to pulmonary hypertension. This is not sponsored yet. Even though we have we have actually 2 proposals pending for review right now. And then relation to the University of Pittsburgh, we are also doing the COVID-19 related research on this one. So we actually have single cell RNA sequencing data from COVID-19 patients, and then we are actually doing we're actually just starting the COVID-19, the RNA sequencing data analysis for the COVID-19 patients. So that's the extramural research. There are a few more under development right now. For example, I just got a meeting request from Harvard.
Kim,
they want to.
You have a minute left.
They're sequencing data about 1,200 sequencing COPD patient data, and they want us to use our own method, computation method, to analyze the patient data to help them with the research. So that's the extramural research, and then we also have some intramural collaboration.
Dr.
So
Kim,
Dr. Youngblood, I think she presented. She was
Dr.
one of the
Kim,
for the data plate.
Dr. Kim, sorry to stop you. We are running out of time. I have one more speaker, and could you please conclude your talk?
Okay, so we got 3 more intramural projects. You can see that what they are, and that's what we're working on the plant, and then goat, and others. And then we have we're using big high performance computing, big data analysis. We do have machine learning capabilities like the Xishuang and others. So if you have any interest in the biological cancer related research, we have a big collaboration. So we can probably bring some of those collaborators for you as well. And then just come to our website. There are a lot of contact information that you can so that you can kind of contact me. You have my email address there as well. So we can kind of discuss what we can do together. So my goal here is to grow together. We don't want to do it there. We don't want to do all of this research all by own. We want to do it. We want to work with you guys together.
Thank you, Dr. Kim. Thank you so much. Participants, you know, you have got a wealth of resources available in the Center for Computational Systems Biology. So for any biological problem, I would talk to Dr. Kim. Thanks again, Dr. Kim. Our final speaker is Dr. Jammieca Mott. We are interested in making STEM into STEAM. So Dr. Mott, it's all yours now.
Good afternoon. I am Dr. Jammieca Mott. I serve as an assistant professor of voice in the Department of Music and Theater. This is actually my first year at Prairie View, coming from Lamar University in Beaumont, Texas. And I am absolutely thrilled to be at Prairie View. Over the years, my main focus in terms of, if you would like to call it research, but expertise, has entailed vocal performance, vocal pedagogy, mentoring or mentorship in underpopulated and underserved areas, and community education. I feel that it's, you know, one's duty to be of service when you've been provided opportunities to go off and pursue your different areas of concentration, and to give back to those who may not be in a similar position in an effort to better themselves. So as a result, I've been fortunate to partner with school districts, the Dallas Independent School District in particular, because before coming to the more southern region of Texas, I lived in Dallas for about 16 years. And so as a result, it provided me an opportunity to go into the schools as a adjudicator first, providing master classes, and then from that point, serving as a vocal pedagogy mentor. For me, I chose to have a different type of balance in terms of the schools that I selected to engage with. And those were schools that serve students who may not be in a position to be provided with applied lessons, whether it was through voice lessons, instrumental lessons, piano lessons, and so forth. And what I always have observed is that our minority students are brilliantly gifted. And sometimes it's simply because of the lack thereof of certain amenities, so to speak. And so as a result of serving as a mentor, it allowed me the opportunity to go in, formulate relationships with the choral directors, as well as the students that I would have worked with from the time they were in the 9th grade up until their senior year, preparing them for UIL activities, whether it be solo and ensemble, choral contest, and ultimately providing those students who were interested in pursuing music studies on the collegiate level. It provided me the opportunity to actually prepare those young people to submit applications and be admitted to those programs, where some of those students were able to actually attend institutions where I served on the faculty. From that point, seeing the success rate of those students as a result of those programs that were formulated throughout that district, and now seeing how some of them have progressed from the undergraduate level onto the graduate level and completing those master's degrees in vocal performance or instrumental performance. Second of all, I think that it's important to recognize that in providing these type of programs, it teaches young people the importance of service. It teaches them the importance of humility. It teaches them the importance of paying it forward. And I think that those are some of the most rewarding things that I have taken from being a part of those type of activities. Because a lot of times when you are in a particular area, you kind of feel like the underdog, but that doesn't necessarily mean that you are not gifted and equipped with the artistic, the artistic brilliance, as well as the intellect. And that's the misconception a lot of times people like to cast over certain areas. So I think that's one thing that I would like to continue to keep as a feather in my cap. And I hope to be able to bring those things to Prairie View and involve current students of mine in non-university professional development activities so that they can get out into the field as students and learn how to cultivate relationships, network, and just learn about the importance of service, accountability, paying it forward, as I've said prior. I think those things are so very important. With those programs, it also has provided students with the opportunity to learn how to create their, as I like to call it, their artistic tribe. They're individuals that they know that they can go to, receive support, receive encouragement, provided opportunities to attend professional venues, those performances, opportunities to establish relationships with professional singers and performers, where they can get a realistic viewpoint of how the field works. I'm a firm believer that the learning process stems far beyond the classroom and a textbook. It's one of those things where it's kind of difficult to take someone somewhere that you haven't been or you don't have a point of reference, so to speak. And so those are things that I am eager and looking forward to being able to hopefully birth and continue in our department. As a result of COVID-19, our field has been affected heavily. I've witnessed dear friends and colleagues of mine, as well as myself, where we have lost a lot of performance venues and opportunities as a result of this horrendous disease. And as a result, we're kind of in limbo. But what I am observing is that it is bringing a community together in solidarity, because it's almost as if we have actually no choice. It's forcing us to become collaborators in a completely different way, compassionate towards individuals, different situations, regardless of what tier of the artistic spectrum that you occupy. And I'm seeing how music is playing a vital role in serving as a therapeutic tool for individuals to be able to cope, and in some instances to survive throughout a crisis. And so as a result, that's something that I've been brainstorming and thinking about, exploring the role and the influence of music therapeutically during a crisis, whether it being focusing on particular composers and certain instrumentation or genres, in an effort to try to dig a little bit deeper and really focusing on the actual role from a psychology standpoint, because they all go hand in hand. They influence one another, especially when we think about the manner in which certain composers write, the material. Yes.
You have a minute left.
Okay, so sorry.
No, it's okay.
So that's a little bit about myself in a nutshell. So I welcome the opportunity to collaborate with individuals if interested. You guys are so brilliant. I don't know how I can come together with some of you guys, but I respect your work, the things that I've been hearing during this hour. But the offer is on the table, and I
Thank you, Dr. Mott.
Thanks.
I have seen many solicitations from National Endowment for the Humanities, and we have social scientists on our campus. We have a College of Juvenile Justice and Psychology.
Yes.
I hope some proposals can be generated as a collaborative effort using music perhaps as one of the additional tools to deal with the stress. I am just thinking aloud. I hope some proposals come along our way. Our office will be very glad to help you in putting together proposals. We very much encourage proposals from non-humanity, or from humanities, non-STEM areas. As I mentioned at the beginning, we want to generate some STEAM proposals too. Even NSF has a directorate for social sciences. So social sciences and social and behavioral sciences, there is a directorate. So we look forward to some collaborative proposals using music as an additional tool in the treatment of stress or depression. I have seen animal therapy. So thank you so much.
Thank you.
It's really a unique experience for us to hear from a musician on this forum. So I very much appreciate your having come forward to deliver a talk. Thank you.
Thank you so much for the opportunity.
All right. Well, Thanks to all of you, presenters and the participants. With that, Data Blitz concludes this session. Again, as I mentioned at the beginning, please feel free to send your comments or suggestions for collaborations through the chat box, and you will receive responses from the presenters by email. And again, I thank you all. Have a good evening. Goodbye now.
Thank you very much.